physiology, 3 more weeks. We're going to finish up the kidney, renal system today, this week. Then you have an exam on Monday on finishing respiration and including the urinary system and then combining them. That's on Monday, and then we'll go into the digestive system and finish up. So you have a problem set this week, but no quiz. The problem set, you have either hard copy, it's on Moodle. It's due on Friday night. If you turn it in earlier, I'll rate it and try to get it back to you earlier. The answer key will get put on Moodle on Saturday morning, so you can't be late. You have to turn it in Friday night. So here's what we're going to do first today. I want to clarify what I want you to understand about the control of glomerular filtration, GFR, because I felt like I kind of botched that on Wednesday. So I'm going to clarify that today. And then we're going to get into reabsorption. So we're moving from filtration to reabsorption, the other key functional element to kidney function. So I don't think I botched this. The idea here is that sympathetic neurons innervate smooth muscle of the afferent arteriole, constriction by GQ mechanisms that you should know. Calcium constriction of the afferent. The afferent arteriole leads to a decrease in glomerular filtration. So that's straightforward, and I don't think I screwed that up. What I do want you to know is that there is this thing called autoregulation, or tubuloglomerular feedback. You get to use words like tubuloglomerular. They're fun to say. Tubuloglomerular feedback, so if there is excessive flow in the distal tubule because of a high GFR, the macula densa senses it by mechanisms that you don't need to know and activates mechanisms that you don't need to know, but leads to constriction of the afferent arteriole. So this is auto-regulation, negative feedback. High flow, constriction, decrease in filtration. That's what I want you to understand. Autoregulation, tubuloglomerular feedback. I also want you to know that there is the converse, but it's complicated. So if there is low flow in the distal tubule, it is sensed by the macula densa, and it triggers renin release. Renin is a hormone that we cover in the other A&P course, but we've got to understand it now because it's an important hormone in the control of the kidney. Renin is released from the juxtaglomerular cells. Remember, the macula densa of the distal tubule is in contact with the juxtaglomerular cells and the afferent arteriole. So the juxtaglomerular cells release renin. Renin triggers a cascade of enzyme events that lead to the production of angiotensin, another hormone, and aldosterone, another hormone. So we will talk about this at the end of the day on Wednesday. We'll talk about this more on Wednesday. Because the effects on GFR, which is what we're trying to focus on, the effects on GFR are complicated. But the effects on water reabsorption through aldosterone are not complicated once you understand water reabsorption, which you don't yet, but where you're going to, and then you will know how to understand aldosterone. So there is this feedback that will become clear by the end of the day Wednesday. And then there is this other point that I do want you to know, that if there is an increase in blood volume, it leads to stretch of the atria because of more volume. More stretch leads to the release of another hormone called atrial natriuretic peptide. That leads to vasodilation of the afferent arteriole, and that would increase GFR. And so the reverse is true of all of these things. We could go, if there is decreased stress and decreased activation of the sympathetic nervous system, you could turn all these arrows upside down and increase GFR. If there is low blood volume, low stretch, you turn all the arrows upside down and decrease GFR. Okay, so remember, things go both ways, even when I'm only telling one half of the story. Questions about this control bit, because we're going to move on to reabsorption. Okay, reabsorption. So I know I've introduced this, and I'm going to go through this quickly. We are going to talk about the reabsorption of water. 99% of the filtered water is reabsorbed. 99.5% of the filtered sodium is reabsorbed. 100% of the filtered glucose is reabsorbed. We're going to talk about those right now. We're going to talk about urea probably on Wednesday. The nephron is responsible for this reabsorption. The proximal tubule, the loop of Henle, and the distal tubule. No, let me do that again. Proximal tubule, loop of Henle, distal tubule, and collecting tubule all have important functions in these reabsorption processes. Remember, we filter it at the glomerulus, and then we process the filtrate to reabsorb solute, like sodium and glucose, and potassium and bicarbonate, and water. And if we don't do that, we would urinate away our life in about 5 minutes flat. I want to remind you that we're talking about an epithelium, the lumen of a tubule, the outside of a tubule lined with these brick-shaped epithelial cells. We talked about this in lab on Thursday and Friday last week. Remember that the epithelium is surrounded by capillaries, and that the apical membrane, this is a name to know, the apical membrane faces the lumen of a tubule. The basolateral membrane faces the interstitium, in which there are capillaries. And we are going to talk about the transport proteins that move solute and water. Well, solute proteins that move solute across the apical membrane and then across the basolateral membrane. And then the reabsorption from the interstitium into the capillaries is passive. There are going to be active transport processes. There are going to be passive transport processes. And there are going to be more passive transport processes. All of these blobs, these ovals, are transport proteins. And we're not going to talk about all of them, but we're going to talk about some of the key ones.
Is the apical membrane present in both the proximal and distal tubule?
The apical membrane is present in proximal, loop of Henle, all segments, distal, and collecting duct. This structure and this cartoon is true for all segments of the nephron. All segments of the nephron have this generic appearance. There are going to be some differences from one segment to the other in terms of what proteins are expressed. So let's talk about sodium. Remember that sodium is a precious commodity, even though it isn't anymore. We evolved in a world where sodium was precious. Getting enough dietary sodium was tricky. and we evolved mechanisms for conserving sodium. It can be counterproductive in today's world when we're living in a world where sodium is easy to get. But that's not the world we evolved in. So you have to remember that we evolved in a world where reabsorbing sodium was paramount. Put yourself in that world in order to understand the kidney. Because if you put yourself in the world of, oh, we need to excrete sodium because we eat so much of it, you're going to get confused. So here's a cartoon. We're going to see a couple of these. Glomerulus. This would be the afferent arteriole. This would be the this is the afferent arteriole, and this is the efferent arteriole. How did I know that? Because the macula densa is in contact with the afferent arteriole more than with the efferent arteriole. This black spot is the macula densa at the beginning of the distal tubule. Glomerulus, proximal tubule, descending loop of Henle, ascending loop of Henle, distal tubule, cortical and medullary collecting tubule. 2/3 of the sodium that is filtered is reabsorbed in the proximal tubule. So we need to understand the mechanisms in the proximal tubule first. Then about 1/4 of the filtered sodium, not 1/4 of what's remaining, but 1/4 of the total, is reabsorbed in the ascending limb of the loop of Henle. We'll talk about those. About 5% is absorbed in the distal tubule, and we probably won't talk about those because the mechanisms here are similar to the mechanisms here. So something has to give, and the distal tubule gives. And it's important. 5% is important, but the mechanisms are similar. About 3% of the filtered sodium is absorbed in the collecting tubule, both cortical and medullary. And we'll talk about those. And if you add this up, I don't know what it adds up to. Maybe not. Somebody do the arithmetic. What does it add up to?
All of that,
All of the yellow boxes.
yeah. 67 plus 370 plus 30,
100. So this is a round-off error, but it's a critical round-off error. So another concept that I want to make clear, because we're going to be using this over and over and over again. When solute is being pumped, which is, we're going to see, solute is being pumped. We're going to be using ATP to pump solute. And if the epithelium is leaky, and by leaky I mean permeable to water, then solute and water are reabsorbed in proportion. The concept here is that water, if the epithelium is leaky, water will follow solute by osmosis. So we pump a solute, and then water will follow it, because water goes from low concentration to high concentration. So if we make a concentration gradient with a pump, water will follow it. And so we're going to see that in the proximal tubule, salt, sodium, and water are absorbed together. But that is not going to be true for other segments of the nephron. So in the proximal tubule, sodium and water are reabsorbed together. And in the ascending limb, it is not water permeable, and we are going to reabsorb sodium but not water, just to illustrate where we're going. Okay, so here is a complicated slide. I gave it to you as a half-page handout because I want you to identify the things to know. There are things here you don't need to know, and there are things you do. And by know, I mean understand. So let's work together on understanding the stories that are told by this image. Okay, let's start here. Proximal tube. An epithelial cell of the proximal tubule. Apical membrane, basolateral membrane. Lumen, interstitial space. Okay? So lumen is yellow, that helps. Interstitial space is blue, that doesn't. Apical membrane is bent, folded, that sort of helps. Basolateral membrane is the rest of it. Okay, everybody oriented? This is an epithelial cell of the proximal tubule. Lumen, interstitium, cells. This is just one of thousands lining that tubule. Everybody with me in these cartoons? Because we're going to see structures like this over and over again for the next 2 days. I mean, today and Wednesday. You need to know that in the basolateral membrane, there is the sodium pump, the sodium-potassium ATPase. So label that on your slide, because that is key. Which way is sodium pumped?
Out.
Out. Sodium is pumped out. Potassium is pumped in, but that's not what we're focusing on. Sodium is pumped out from the cytoplasm into the interstitial space, and it's pumped into the interstitial space because it's expressed on the basolateral membrane in contact with the interstitial space. It is not expressed on the apical membrane. There are all kinds of cell biology that target the expression of this protein on this membrane. You can get back into the cell physiology of how, membrane proteins are synthesized and co-translationally, never mind. There are lots of control mechanisms that put this protein in this membrane. Okay, now let's look at the apical membrane. Let's look at this one here, a sodium proton exchanger. A sodium proton exchanger. So, sodium moves down its concentration gradient in exchange for protons being pumped out into the lumen of the tubule. All right, we just made a path for sodium reabsorption. You should see that we've moved sodium from lumen, from luminal fluid, from urinary fluid. It's not urine yet, but it's going to become urine. Urinary fluid into the cell, and we've pumped, and that sodium is then pumped into the interstitial space, where it is then passively reabsorbed by capillaries. So we have just made a path that pumps sodium from urinary fluid into the blood. And it used ATP. It required energy. All right, that's not the only pathway. But that is, the concept is right there. Everybody with me on this concept of using ATP to pump sodium. We have another transport protein called the sodium glucose co-transporter. Sodium and glucose are co-transported from the urinary fluid into the cytoplasm. We're going to focus on glucose in a few minutes. Right now we're focusing on sodium. Sodium is pumped by two mechanisms. Well, two mechanisms on the apical membrane and one mechanism on the basolateral membrane. Sodium also on the basolateral membrane moves in response to a sodium bicarbonate co-transporter. In this case, right, so you can see sodium is moving out. So sodium is moving uphill. Energetically, we're moving from a low concentration of sodium to a higher concentration of sodium when we move from cytoplasm to outside. This is using, this is not using the gradient. It's actually working against the gradient. Sodium, in this case, bicarbonate, is driving, bicarbonate is driving the flow of sodium. But there are going to be other cases where sodium drives the movement of bicarbonate. The co-transport, the concentrations matter, but in this case, it's another path. So I do want you to remember that there is a sodium bicarbonate co-transporter on the apical membrane. So you might be saying, well, he's circling everything. Well, I haven't circled everything yet, and I'm almost done. Those are the mechanisms for sodium pumping in the proximal tubule. Here is the thing I want you to know about water. Water flows passively because this is leaky to water. And I need to introduce two terms. Sodium is pumped transcellularly. Transcellular pumping through the cells, transcellular. Water moves paracellularly. Paracellular transport of water. Water is going around the cells. See how it's not going through the cell, it's going around the cell through these junctions. These junctions in the proximal tubule are leaky, and that's why water can follow the pumping of sodium. If I didn't circle it, you don't need to know it. Introduction?
The bicarbonate sodium co-transporter, it still needs energy for it to function, right?
Yeah.
So is the energy coming from the charge of bicarbonate?
It's coming from the bicarbonate gradient.
Gradient, I see.
So there are some things I'm not circling, but you probably already, you know that bicarbonate comes from carbonic anhydrase. So when we start to talk about bicarbonate in a few minutes, we'll need to focus on this. But for sodium, let's just do it like that. Question, yes?
Did you repeat about the transcellular and paracellular?
Sodium is moving through the cell. And so we call that transcellular. Water is moving around the cell through these junctions outside of the cell, and we call that paracellular.
But sodium is also moving.
Well, yeah, I suppose. Just not worry about it. Because sodium is also doing this tricky thing here. So we're not going to talk about sodium paracellular movement because it has canceled out. We're going to talk about water paracellular movement. Okay? So if on an exam I ask you for sodium transport mechanisms and you include sodium paracellular movement, fine, I'm not going to take off because you included something right. But it's not the key point. Other questions? Okay, let's go over to the thick ascending limb. So we're moving from here to here. Remember, I'm not focusing on thick or thin. So we're just focusing on the ascending limb. But it happens to be the thick segment of the ascending limb. We've just talked about the mechanisms for 2/3 of the sodium reabsorption and 2/3 of the water reabsorption. And now we're going to get into a mechanism for 1/4 of the sodium reabsorption, but no water reabsorption. concepts are, now things are gonna move faster now because the concepts are the same. A sodium-potassium pump, the sodium pump is pumping sodium. A sodium proton exchanger, same thing as over here. So a commonality between, two commonalities between the proximal tubule and the ascending limb of the loop of Henle. Now, a twist. Another transporter on the apical membrane that moves sodium into the cytosol. And it has a very helpful name. It's called the sodium-potassium 2-chloride cotransporter. This is one time where the physiologists really do us a favor. They named it for what it does. So if you can name it, you know what it does. If you know what it does, you can name it. The sodium-potassium 2-chloride cotransporter, or NAK2Cl cotransporter. I almost always write it like that. NAK2Cl cotransporter. So again, we have mechanisms for sodium movement across the apical membrane and pumping out the basolateral membrane. This transporter, the chloride bicarbonate exchanger, is going to come in important when we talk about bicarbonate, but it's not essential for sodium understanding. Okay, so again, mechanisms for solute, in this case sodium flow, sodium being pumped. Notice this. Very low water permeability. We don't see water permeability here either. We're not going to worry about sodium paracellular diffusion. We're just not going to worry about it. It's not key for this. So we have the reabsorption of sodium from urinary fluid back into blood, but no water movement. We're going to see that that's important when we talk about water in a few minutes. Okay? Distal convolute, any questions about proximal ascending limb of the loop of Henle? Now, let's go, we're going to go to the next segment. Proximal ascending limb. We're going to skip, I think we're going to skip the distal tubule. Oh no, we're not going to skip the distal tubule, we're going to cover it right here. The distal tubule, same themes. Sodium, potassium, pump, the sodium, potassium ATPase, pumping sodium, and a sodium chloride cotransporter. Sodium chloride cotransporter and the sodium potassium 2-chloride cotransporter. This cotransporter and this cotransporter are not the same proteins. They have different structures, they have different pharmacology. They're blocked by different drugs, is what I'm saying. But their physiology is similar. Sodium moving down its concentration gradient, being pumped out. Sodium moving transcellularly, water not moving. Solute, sodium transport, no water transport in the distal tubule. And finally, so now we're going, we did the distal tubule, now we're going to do the collecting tubule. But it gets, we're going to go fast. Sodium potassium pump on the basolateral membrane, pumping sodium out. a sodium channel. Not a co-transporter, it's a channel. It's a passive channel that allows sodium to flow down its concentration gradient, and sodium is pumped out. Transcellular. This channel has a helpful name. It's called the epithelial sodium channel. You can call it ENIC, because everybody else does. So we have just in terms of names, the sodium ATPase, which you can call the sodium potassium ATPase, or you can call the sodium potassium pump, or the sodium pump. Sodium glucose co-transporter. Sodium proton, or NaH exchanger. Sodium potassium 2 chloride co-transporter, NaH exchanger, sodium pump. Sodium pump, NaCl co-transporter. Sodium pump, ENaC. What does the E stand for? Epithelium, because it's not expressed very much in neurons, where there's also voltage-gated sodium channels in muscle cells. ENaC is not expressed very much in those cells. Questions about sodium transport? We've done it. So I like this cartoon because, let me just lower it so we can see it. I like this cartoon because it's the picture that tells the story. And if you can narrate the picture, you can tell the story. And if you can tell the story, you can tell the story to me, which tells me that you know the story, which is a good thing to be able to do. These are the words that are the caption to that figure. So I'm not going to go through this because I've said all these words. But if you like words, look at the next slide. But if you like pictures, look at your annotated picture. And I think you should look at both, actually. But I'm not going to spend time on this because I just said all these words. Did I give you this slide as a handout? Okay, let me just annotate it. Sodium potassium ATPase, yeah, we got to know that. Sodium glucose co-transporter, yes. Sodium proton exchanger, yes. Paracellular diffusion, you can cross this out, right? Because I crossed it out on the slide before, so I should have crossed it out here. So just cross this out. I want you to know about paracellular diffusion of water. So just, I'm really tempted to screw up the screen, but I'm not going to. Just cross out paracellular diffusion of sodium AN. Just cross out sodium. I want you to know about water moving paracellular. Okay, thick ascending limb. We have the sodium potassium 2 chloride co-transporter. I want you to know that there is no water paracellular movement in the thick ascending limb. I don't care whether you know about sodium. Distal tubule, we have the sodium chloride co-transporter. Cortical collecting tubule, we have ENaC. Okay. Questions? Questions about my annotation of this slide that I should have annotated previously? So,
is there a reason why the bicarbonate one isn't in the proximal?
No, there's no good reason why the sodium bicarbonate co-transporter is not in this list. It should be.
Okay.
So add sodium bicarbonate. It's a cotransporter in the basolateral membrane. Sodium bicarbonate cotransporter in the basolateral membrane of the proximal tubule. To that. Other inconsistencies? Questions? Okay, so remember, we've talked about transcellular pumping of sodium, paracellular movement of water in some segments, but not all, and then passive reabsorption into the blood that is surrounding the tubule. And you should be thinking that in a leaky epithelium, like the proximal tubule, water follows solute. Pump solute, and you're in a leaky epithelium, leaky to water, water will follow solute. So now let's move on to glucose. Using the same themes that we've already developed for sodium. All of the glucose is reabsorbed in the proximal tubule and the very beginning of the loop of Henle. So we can simplify this and just say all of the glucose is reabsorbed in the proximal tubule. Because this part of the loop of Henle is so close to the proximal tubule, we're just going to make it an honorary member of this proximal tubule for glucose. So we only have to think about one segment of the nephron for glucose reabsorption, the proximal tubule. And the mechanisms are similar. In fact, one of them has already been mentioned. Two of them have already been mentioned. Sodium potassium ATPase sets up a sodium gradient, low concentration of sodium on the inside. Sodium and glucose. Now we're focusing on glucose. Glucose is pumped from urinary fluid into the cell, and it flows via passive uniporter into the interstitium where it is picked up by blood. So for glucose, we do not have a glucose ATPase. We use the sodium ATPase to build a sodium gradient, and then we use the sodium gradient to pump glucose into the cell, and then it flows down its concentration gradient into the interstitium. These transport proteins have a name, sodium glucose transporter, SGLT. I am not going to ask you to know the difference between SGLT1 and SGLT2, but I do want you to be able to articulate the difference between a sodium-dependent cotransporter that is using the sodium gradient to pump glucose and a glucose uniporter that is simply passive diffusion. So these are called sodium glucose cotransporters. You can use SGLT as the abbreviation. These are glucose uniporters, glucose passive transporters. Nothing active about this. It's not a channel, but it's a transporter. And they're called GLUT 1 and GLUT 2. I am not going to ask you to know the difference between GLUT 1 and GLUT 2. I'm not going to ask you to know the difference between SGLT1 and SGLT2. I am going to ask you to know the difference between SGLT and GLUT. So this slide, articulates a difference. The key difference is that as we get to the late proximal tubule, we have mechanisms for picking up glucose at very low concentrations, so that by the time we are leaving the proximal tubule, the tubules have grabbed and reabsorbed all the glucose that was filtered, as long as the transport processes were not exceeded in their ability to transport. We're going to talk about that in a moment. This is the mechanism for glucose co-transport. For glucose reabsorption, is what I meant to say. And 100% of it is done in the proximal tubule. If the concentration of glucose in the proximal tubule is high, because of, let's say, diabetes. Diabetes is high blood glucose. So high blood glucose would lead to high filtered glucose. If the filtered glucose exceeds the capacity of SGLT1 and 2 and GLUT to move it, then some of the glucose will get past the proximal tubule. And if it gets past the proximal tubule, it gets all the way into urine, and that's why diabetics have glucose in their urine, unless it's controlled. So let's do some clickers. Actually, let's take a, well, that's not a clicker, that's just the words. Sorry. That's the words that describe the story that is illustrated on the previous slide. Sodium-dependent co-transport, active transport on the apical membrane, passive GLUT transporters on the basolateral membrane. Passive diffusion into the bloodstream. Reabsorption of glucose. Questions? The next slide, which I can't remember, did I give you this one? Yeah. This is just an illustration of if plasma glucose concentration exceeds about 250, so normal plasma glucose is about 80 to 100. If the glucose concentration in plasma is 250 because of diabetes, then the ability of the proximal tubule to reabsorb it is exceeded. And now we start to get some excretion. That's all that this slide is saying. I will not ask you to interpret data like this. If the plasma glucose is high, the ability of reabsorption falls below the amount that is filtered, then we get some excretion. And that's why diabetics excrete glucose if their diabetes is uncontrolled. And that's why urinary glucose is a marker for diabetes. Okay, so now let's do some clickers. Let's take a 10-minute break. I've been talking for 45 minutes or so. Let's take a 10-minute break. There's a series of clickers. I believe I gave them to you on your handout. There's just 1, 2, 3, 4 clicker questions on 3 slides. So go ahead and work through them. Then we'll reveal the answers, see if there are any questions, and then move on to the next subject. Okay?